All right, folks, for Chapter 3, I think I've got a pretty good list of videos here for you. Although for Chapter 3, one of the things I wanted to focus on that sometimes students have trouble with is this idea of diffusion and osmosis, kind of what they are, how they work, the idea of tonicity, and how it works, what these definitions are. And so let me kind of quickly go through that, and then I think the active transport kind of falls in place after you understand passive transport. The other sections, not that the other sections of the chapter aren't important. However, when you're talking about prokaryotic and eukaryotic cells, it's really just kind of comparisons. So what is it that a prokaryotic cell is versus a eukaryotic cell? Prokaryotic cells don't have membrane-bound organelles. They don't have a nucleus the way eukaryotic cells do. Sometimes prokaryotic cells are termed as more simple. I don't really like that because they're complex in and of themselves. But just some comparisons there. Prokaryotic cells are usually smaller than eukaryotic cells, and so you can work your way through some of the differences and really just what the organelles are. This is just some memorization. A good idea for this would be to draw this out and just relabel it. That's a good way to kind of store this stuff. One thing students do kind of get confused on here is both plant and animal cells are eukaryotic cells, right? So both plant and animal cells are eukaryotic. It's bacteria that are prokaryotic, right? And then just look at some of the differences between plant and animal cells. For instance, plant cells have chloroplasts because they need to photosynthesize. Typically, plant cells will have a large kind of vacuole here. That helps plants stand up. Gives them some what's called turgor pressure, presses against these cell walls. Typically, plant cells will have cell walls where animal cells won't. And so those are just some of the differences to look at. And then I think I've got some videos that are pretty good with some of the cytoskeletal stuff, right, in the endomembrane stuff. So let's look at passive transport. The cell membrane is selectively permeable. And so what that means is some things can move across the membrane quite easily and some things can't, right? So it's kind of like my door here in my office. If I have the key, I can come in. If you don't have the key and the door's locked, then you can't come in, right? And so the door in that sense is selectively permeable. Membranes typically allow things like water or things that are it depends on their charge, right? And it also depends on how large they are. So really large proteins aren't going to move across membranes very easily. And if you understand what membranes are composed of, so I don't have my stylus, so I'm drawing with the mouse here, so bear with me. But if I have, right, so remember these are the phospholipids that make up cell membranes, right? And so if I have them side by side, whoops. And let me close that in. Right, and then that makes up a membrane. I mean, look at that drawing, right? That's spectacular. There are spaces in here, right? There's a little space there. There's a little space there. And so things can move through that space either in or out. Depending, right? And so if I have like a really large, let's say I have a really large protein out here, right? That protein's not going to move through that space. It's just too big. It needs a gate, and it's probably going to need ATP in order to do it. However, if I have a really small particle, right, out here, and let's say its charge is correct, it can move through that space relatively easily. And so in that sense, membranes are selectively permeable, right? They'll allow certain things to move through, whereas other things they might not. Diffusion is just simply the movement of molecules down a concentration gradient. So a movement from high concentration to low concentration. And the thing about it being passive is that it requires no energy to do so. And so if I look at this membrane setup, I have a lot of a substance on one side of the membrane and relatively little or none on the other. And so assuming that this membrane allows these things to pass through, they will pass through, right, because of that concentration gradient. So let's say that the membrane allowed sugar to pass through, and let's say that's what these molecules were. these molecules of sugar will move from this high concentration of sugar to this low concentration of sugar across that membrane. That's diffusion. Another way to think about diffusion is, this is not exactly diffusion, but it works. The idea, if I have cologne and I spray it here, right, I'm going to smell it first, and then if I'm standing in a room, people at the back of the room will smell it last, right, because it diffuses out. Think about putting a drop of food coloring in a container of water, right, really high concentration of food coloring where you dropped it. If you leave it there for an extended period of time, eventually it diffuses out and all of the water becomes one color. And so that's diffusion, just a random movement of molecules down a concentration gradient from high concentration to low concentration. And they'll keep moving until they reach this kind of equilibrium, right? That's diffusion. The important thing to remember about diffusion is it's a passive process. It requires no energy, it requires no ATP, right? So then if we look at osmosis, right, the difference between diffusion and osmosis is like the difference between saying dogs and beagles. All beagles are dogs, right? But all dogs aren't beagles. Osmosis is just a specific type of diffusion. It's the diffusion specifically, right? Osmosis is the diffusion of water, right, across a semi-permeable membrane, right? And so what they're showing you here is, let's say I have a beaker, and in that beaker I have a membrane, and that membrane is permeable to water, but let's say it's not permeable to anything else, so nothing else can move across it. And so also what you'll see, let me do this. This is probably a good place to do so let's say, and I'll use black, let's say here I have a beaker, right? And let me talk about concentrations of liquids first. So let's say in this beaker I have a 30% concentration, and let's just call it salt water, sodium chloride. I hate drawing with a mouse. And let's say in this beaker I have another fluid, and let's say it's 10% sodium chloride. Right? Let's talk about some terms first. If I compare these two liquids to each other, I can talk about their tonicities, right? And talking about tonicity, using the terms I'm going to use, those terms are relative, right? So it would be like saying this pencil is big. Well, compared to what? Right? I can't just say it's big and leave it at that, because there's no frame of reference. Same thing with these terms I'm about to use, hyper, hypo, and isotonic. They are all relative terms. So what it means to be hypertonic is that you have a higher solute concentration compared to another solution. Hypotonic means you have a lower solute concentration, right? So if I look at these two, and I talk about this as being fluid A or beaker A, and that being beaker B. Beaker A would be the hypertonic, H-Y-P-E. This is really bad. I might just put a word box in here, but whatever, it's comical at this point. This would be the hypertonic solution because it has a higher solute concentration. This is the hypotonic solution because it has a lower solute concentration. Notice I'm comparing them to each other, right? I couldn't just ask you, is A hyper, hypo, or isotonic if you didn't have another solution to compare it to, right? The other thing I could ask about these solutions is, if solution A is 30% salt, how much water is it? Well, it's got to be 100% total, so if it's 30% salt, it's 70% water. If beaker B is 10% salt, how much water is it? Well, it's 90% water, right? Because if it's 10% salt, it's got to be 90% water. 90 plus 10 gives you 100, right? And so if the concentrations were the same, then they would be isotonic, right? So hyper, higher concentration of solutes, hypo, lower concentration of solutes, right? Now, what would happen if, let's say I had a contraption, whoops, I gotta do this. Let's say I had a tube, and the tube looked like this, right? And let's say the tube was full of water, equal on both sides, and let's say I put a semi-permeable membrane in the middle of the tube. Water can cross that membrane, nothing else can, only water. Let's say this side is A, this side is B. This side has a 30% concentration of salt in it, and this side has a 10% concentration of salt in it. And what I want to know is, since both of these columns are filled completely to the same level with water, and since this membrane is permeable to water only, what would happen to the volume in these columns, right, over time? Well, since this side is 30% salt, it's going to be 70% water. And since this side is 10% salt, it's going to be 90% water. And if you remember what I said before about diffusion, diffusion moves from high concentration to low concentration of whatever you're talking about. So here, since I'm talking about water, the water would want to move from a high concentration of water. Right, let me do this in blue so you'll see. It's going to want to move from a high concentration of water, right, to a low concentration of water. Right, 90% is a higher water concentration than 70%. Right, and at first you have to kind of think about that a little bit, because usually when we talk about concentrations, we're not talking about water. But here, since the only thing across that membrane is water, that's the only thing that can move across it, then you have to think that way. Right, so 90% water, 70% water. So what that means is water is going to move from this direction across. So the water here, this level is going to go up, and this level is going to go down. Right? And so it's going to keep moving that way until these two concentrations, or even the water concentrations, are equal. Right? That's osmosis. Just the movement of water down its concentration gradient across a selectively permeable membrane. Right? No energy is used. You're not using ATP. This is how water moves in your system daily. It's why when you drink, so if you wake up in the morning and you don't usually drink a whole lot of water, you drink a glass or a glass and a half of water, before long you're going to the bathroom. Why? Because the water is diffusing across. It ends up in your bloodstream. It ends up in your kidneys, and then you end up urinating. Right? That's osmosis. Just a specific type of diffusion. It's the diffusion of water down a concentration gradient across a semi-permeable membrane. Right? There's the terms for tonicity. How does this kind of work internally? Well, if you take red blood cells, for instance, and you put them in salt water, so let's say you injected salt water right into your vascular system, because that concentration of salt water would be hypertonic compared to what's already in you, it would tend to pull water out of those red blood cells. Right? So think about that. Right? Hypertonic solution outside of the red blood cells, it's going to want to pull water out of those red blood cells and they'll shrivel. Right? Not good for you. The opposite is true though too. So let's say I inject pure water. So when you go to the hospital, if you need fluids, they're not putting pure distilled water into your system. Right? That would be a huge problem for you. What they're putting into your system is usually physiological saline, so about 0.9%. If they were to put pure water into your system through your bloodstream, right, then your red blood cells would swell, and because they don't have cell walls, they would rupture, right, called hemolysis. Now, that doesn't mean that you shouldn't drink pure water, right? Because when you consume regular water, you've got enough salt concentration, and you've probably eat a potato chip or whatever. And so when you eat or when you drink water, even if it's distilled water, it's not a problem for you because you have enough electrolytes in you already. But if you were to inject it straight in, then that could be an issue. The other time drinking water might become an issue. One, if you drink way too much, right? And so kind of a common buzzword now, especially in the dieting world, are things that you should avoid because they're toxic, right? So you hear people talk about sugar being toxic, right? Things are toxic based on the dose that you get, right? Sugar is not toxic. If you ate 5 pounds of sugar in one sitting, probably toxic. Same thing's true with water, right? So there was a radio show that did a contest years ago, and what they were doing is seeing who could drink the most water without going to the bathroom. And what happened is the lady ended up dying. Why? Because she drank so much water, she didn't go to the bathroom when she should have. It got her electrolytes really out of balance. Now she had all this water out in her system, and her tissues started to absorb it, started to pull it in, right? Same way that's happening here in these red blood cells. And so as her tissue started pulling it in, her brain started pulling that water in, and her brain started swelling. When your brain swells, it puts pressure on your brainstem, you lose consciousness, and then if it's bad enough, you lose your life, right? And so water, there is a such thing as a water toxicity, and that's it, right? Sometimes even pets can have this. So if you take your pet, so sometimes people will take their pet maybe to the river or to the ocean or whatever, and pets will spend a lot of time just splashing around, and maybe they chase a ball into the water a lot and they consume a lot of water. Or sometimes if they're acting weird after that, it could be a water toxicity issue. So you kind of have to be careful of that, right? But that's kind of the role or the issues that it can play in us. Kind of less problematic if you come home, right, and you have plants in your house and they're wilted. The reason they're wilted is, if I go back up, now that we understand that concept, if I go back up and look at the plant cell, the reason the plant cells are wilted is because all of the water has left the vacuole, or a lot of it has left the vacuole. And what happens is, inside, I wonder if I can do this. Let me copy that image and paste it over here so I can draw on it. Are you going to let me? Yes. Although that looks horrible. Let me do this. Let me new slide that. And let me cut and paste that over here. So when the plant wilts, what's happening is all of the water has left that vacuole. Typically what happens when a plant vacuole is full of water, it actually pushes against that cell wall in all directions, right? And in that sense, it gives the cell, think about a water balloon in a box, right? If you filled it up, it's going to push on the box and give the box some rigidity. Well, as you lose water, you lose this pressure. It's called turgor pressure against the cell wall. And so if it's not a woody plant, the plant will then wilt, right? You give it water and it stands back up. Why? Because the cell vacuole, or that central vacuole, fills back up with water. It pushes back up against that cell wall and causes it to stand back up. And that's all osmosis, right? That's all an osmotic force. And so you see it in action every day. You just don't realize that you do. Right. All right, did I miss anything else in tonicity? Nope, I think I got that. Oh, I forgot it. That's what I just covered. Right, I forgot that little diagram was at the bottom. Right. Whatever, my drawings are better. The other thing they talk a little bit about here is facilitated transport or facilitated diffusion. So a facilitator is anything that helps a process move along. If I go back maybe to my drawing, or even here, or even this, let's say that there was a little gate or little channels in here that helped the water move. Not helped in the sense of gave it energy to move, but maybe the holes were just a little bit larger so water could move through more easily. Right? That would be facilitated diffusion. Anything that helps the process along, or facilitates the process, that's facilitated diffusion, right, or facilitated transport, right? And then if we looked at active transport, that's anything that requires ATP. Think about a pump, right? Pumps are active, right? Anytime, so up until this point, I've talked about moving things from high concentration to low concentration. So think about moving things down a hill, right? You give it a little tap, and away it goes. If you want to do something active, right, so maybe you want to move something up its concentration gradient. So I want to move something, maybe I want to move, say I want to move this potassium from here over to there, right? Well, the problem is there's so much potassium over there, this one by itself doesn't want to go over there. And so the only way it's going to go over there is if I pump it over there through an active transport system or use ATP, right? So move things against their gradient, right? And so that could be any number of things. It could be a gradient, so just a salt gradient, for instance, so sodium chloride, sugar gradient, right? Or it could be a charge gradient, right? So notice that here they're showing you positives and negative charges too. So if I have a bunch of positive charges on one side and a bunch of negative charges on the other, right? Well, if I want to move more positive charges to the positive side, then that's going to require energy to do that. And I'm going to have some kind of active transport system in which to do that, right? All right, I think that's probably enough of an overview of those. Obviously, if I've spent some time on them, I'm probably going to ask you some questions about diffusion osmosis. It's just a huge concept, especially in biology, right? So if you have any questions, please let me know.